Hello students, the next lecture over here is the classification of enzymes. A very interesting and very simple lecture. A systematic nomenclature and classification of enzymes is based on the types of reaction that the enzyme carries out. On the types of reaction that the enzyme carries out, we classify the enzymes. And then number one. Number two, on the basis of the substrate on which the enzyme acts. A systematic nomenclature of enzymes and classification of enzymes is based on the reaction types and the reaction mechanism, which is given by the International Union of Biochemistry in 1961. On the basis of this, all the enzymes have been classified into 6 groups. How many groups? Into 6 groups. These are the 6 groups. Oxidoreductases, very easy. Oxidoreductases, something to do with oxidation and reduction. Transferases, something to do with transfer of something. Ase is always written at the end of most of the enzyme. Hydrolases, something to do with water. Ligases, right? Isomerases, isomerism, ligases, ligation, joining, liases, lysis, breaking down, transferases, transferring from one place to another, oxidoreductases, oxidation and reduction, transferases, transferring, hydrolases, something to do with water addition or removal, liases, lysis, breaking down, isomerases, preparation of isomers, ligases, something doing with ligation, something doing with joining, very, very easy concepts. Oxidoreductases. Like oxidoreductases, what do? These are enzymes that catalyze the oxidation and the reduction of the substrate and act by removing oxygen or by adding electron or hydrogen to the substrate from or to the substrate. For example, cytochrome oxidase oxidizes cytochrome. Dehydrogenases and oxidases are examples of oxidoreductases. What do? They bring about oxidation or reduction of the substrate. How? By either adding or removing electron or hydrogen from the substrate. Example, very common example, dehydrogenases and oxidases, and then accept that cytochrome oxidase, which oxidizes the cytochrome. I have just told you in the previous lecture that cytochromes are present in the electron transport chain. Oxidoreductases, transfer of oxygen and hydrogen atoms between the substances in redox reactions. Redox reactions are those reactions that are simultaneously taking place at the same time. Transferases. Transferases, transfer of a chemical group from one substance to another. For example here, transaminases. Transaminases, phosphorylases. These transfer groups from one point to another. They transfer chemical groups from one substance to another. They take a group and transfer it totally from one substance to the other. For example, AB plus C. AB plus C. AB plus C in the presence of enzyme. C will be transferred to B. A is going to be left alone. A plus CB. AB plus C. CB is going to be transferred to C. This is the case in transferases. Transaminases. Amino acid is going to be transferred from one point to another. Phosphorylases. Phosphate is going to be transferred from one substance to another. These are the enzymes that will carry out the transfer of phosphate groups. This is the group that is going to bring about the transfer of aminases. Hydrolases, hydrolysis, breaking down in the presence of water by adding water, hydrolysis. Peptidases, breaking down of peptide bond by addition of one hydrogen and OH. Lipases, breaking down of lipid bond, the ester bond. Amylases, breaking down of what do we call it? Breaking down of the bond that is present between the carbohydrate. What is that? Glycosidic linkage. By breaking down the glycosidic linkage adding hydrogen to one member and OH to the other member. These are hydrolases, hydrolysis reactions which are taking place, ligases. Addition or removal of a chemical group in the absence of water. Hydrolysis in the presence of water. Ligases work in the absence of water. Decarboxylases, removal of carbon dioxide. Dehydrogenases, removal of hydrogen. Ligases, removal of a chemical group other than hydrolysis. Matlab water is not going to be involved in this. Hydrogen and OH will not be added in this situation. Isomerases, rearrange groups within a molecule. Example, isomerases in the glycolysis reaction. Hexokinases rearrange the groups from glucose and convert them into fructose during the glycolysis reaction. That is an example of isomerases. That is an example of isomerase in which intermolecular rearrangement of atoms takes place. These atoms, they form isomers of one another. For example, phosphohexokinase isomerase. It converts glucose 6-phosphate to fructose 6-phosphate. Then the next one that we have are the ligases. Ligases, these are enzymes that bring about joining of two molecules. The energy for this is provided by the ATP. It is provided by ATP. For example, polymerases, responsible for linking up polymers of RNA and DNA. Synthetases, which are going to synthesize various compounds in the presence of ATP. Two bonds are going to be formed. And ligases, DNA ligase is an enzyme which is very, very important, and it is present in the DNA during the formation of the replication of DNA ligases. Now, this was all regarding the classification of enzymes. The last topic of this chapter is the control of the metabolic pathways. The cells contain organelles which have enzymes and bind to their inner membranes. Organelles have varying conditions to suit the enzymes that they contain. By controlling these conditions, the enzymes available in the cell, they can control the metabolic pathways that are present inside them. The cells can make use of their own properties, enzyme properties, to control the metabolic pathway. For example, the concentration of one product can reduce the speed of the reaction. Now I will go to the board to explain this concept to you. You can give me questions if you have. Negative feedback. A by enzyme A is converted into B. B by enzyme B is converted into C. C by enzyme C is converted into D. And D by enzyme D is converted into E. Now, what happens? That this E is going to move back and it is going to come to the A situation again. It will come here and it will come and act as an inhibitor for the enzyme in place of A. And what will it do? It will stop the ongoing reaction. This is the final product. This is called negative feedback mechanism, in which the action of the enzyme is controlled by the end product of the whole pathway that is taking place. This is a series of reactions that is taking place inside our body, inside the metabolic pathways. Let me go to the board and let me explain it for you people so that it can be easy for you people to understand, and then we will call it a day. Okay. Enzyme inhibition was a previous lecture that we studied. Let me just rub it off. Okay, now enzyme A, substrate A, worked upon by enzyme A is going to produce B. B going to be converted into C. C going to be converted into D. Now this is a metabolic pathway. Now this one, what happens? This D, the presence of D is going to give A the enzyme working over here. Now, if this is the enzyme that is working over here, this is the enzyme that is working over here, this is the enzyme that is working over here, this D is going to come over here and it is going to cheat this enzyme. It is going to attach to the allosteric site of this enzyme, and it is going to bring about the non-competitive inhibition, and it is going to stop this enzyme from acting on A, and therefore it is going to control the reaction. When will this happen? When the required amount of B has been produced. This is the negative feedback mechanism. Our body mechanisms are mostly controlled by the negative feedback to prevent excess of products, excess of materials produced inside the body. We have examples of positive feedback in our body also. For example, the childbirth impulse is a positive, but positive feedback takes place only in some situations. This feedback in which the final product, let me write it down on the board. This feedback in which the final product is inhibiting the first enzyme. It is inhibiting the first enzyme. It is going to stop the reaction. It is going to control the rate of the reaction. It is going to control the rate of the reaction. This is called the feedback inhibition. What do we call it? We call it the feedback inhibition. And this is actually a negative feedback mechanism. It is the negative feedback mechanism. It is the negative feedback mechanism in which it is going to control the rate of the reaction. This is what is taking place normally in all the body situations. It is normally taking place in all the body situations, right? I think this is all for the topic today. Take care of yourself. See you people next time, and do join my channel. Do recommend your friends to come to this channel and take good lectures, easy lectures in biology, concepts made very, very easy. I am not going into detail because I need every student to get the basic concept of biology. Once you understand the basic concept, you can look for the detailed concept. You can contact me for the detailed concept. I will give you detailed lectures whenever required. But at present, I am just introducing the basic concepts that are required at college level. See you people next time. Keep working hard, keep studying. Best of luck. Recommend the channel to your friends and see me next time again. Come to my channel, attend more lectures. Allah Hafiz. Best of luck.